Welcome to Lab 4 of Bio 206. So once again, I'm going to ask that you make sure you have your lab packet open to follow along, and then your lab manual as well. We're going to be in exercise 12 as well as exercise 14 of your lab manual. So to get started with today's lab, let's start running through these objectives. So number 1 says, identify in a diagram, define the following terms used to describe ultrastructure of a muscle. So as we look at page 184 in your lab manual, you'll see this is looking at muscle tissue, an actual photomicrograph, then this shows you a nice illustration. What's really nice is it shows how the muscle fiber, so remember, a muscle fiber equals actually one muscle cell. And as we look at this ultrastructure of the muscle cell, you can see that it's divided into small myofibrils. So when we look, that's one myofibril. You can see many, many, many within that muscle cell. As we look at one myofibril, you'll see that it's divided into several structures called sarcomere. Now, I really like this picture illustration in your book because when we teach sarcomere, it's so hard to envision how those sarcomeres are going down. So you can see in this picture, sarcomere, 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 you can see that they're going the whole length of the myofibril. They go the whole length of each myofibril within the whole muscle cell. As we look at the sarcomere structure, you can get a little clearer picture of it, but as you can see, there's lots of different terminologies associated with it. So we're going to take a little time to look at that a little bit more detailed. So this illustration of the sarcomere introduces you to the organization of our myofilaments. And our myofilaments are going to be actin and myosin. And remember, actin is going to be thin. Okay, it basically spells the word thin, while myosin will be a much thicker fiber. So we learned sarcomere structure. I think the best way to learn it is to actually take time to draw it out. So let's do that. So when we look at sarcomere structure, I like to start in the middle. Wrong pen, sorry about that. Start in the middle. So this structure here, this is going to be the middle of our myosin. And this structure is the M line. So the M line is the middle of the myosin. It's also the middle of the sarcomere. Branching off of our M line are going to be myosin fibers. Okay, so notice the myosin is braced in the middle of our sarcomere on either side of this M line. Now the next myofilament, our thin filament, is actin. And you can see it's going to overlap the myosin a little bit. But when we look at actin, it's braced on this structure right here, known as the Z line or the Z disc, or spelled with a K. I've seen it too many different ways. And you'll see that just like myosin, actin will branch off the Z line in both directions. So notice actin is on both sides, and we have a Z line. When we look at the actual borders of the sarcomere itself, the sarcomere runs Z to Z. Okay. So then the question becomes, what is that other actin? Well, remember, we had sarcomere next to sarcomere next to sarcomere in that other picture. So this is sarcomere number 1. This is sarcomere number 2. Then these would be actin for sarcomere number 3. Okay? So remember, this is myosin. And then this blue, that's going to be actin. Now there are certain zones and areas that we can see. So here in the middle, Where there's only myosin, this is called the H zone. So when we look at the H zone, it's the middle of the myosin, but there's no actin. However, on either side, oops, wrong color here. Let's switch colors. On either side, this is known as I call it the ZOO, but ZOO stands for zone of overlap. Okay, so all of this right here, where actin and myosin overlap, that's the zone of overlap. Now there's some other terms associated. They're called bands. And when we look at the bands, the A band goes from end of myosin to the other end of myosin. So notice it composes the H zone and the overlap, where the I bands, this is going to be only actin. So this also is an I band. So as we look at these structures, drawing it out I think really helps to get the different colors, get your colored pencils, markers, crayons, whatever, and draw the different colors so you can start to see this is what overlaps with this. What happens in muscle contraction, and you'll see these in the videos that are posted, is myosin is going to pull actin this way. So what happens is this H zone it drops all the way to like this, and then this whole structure becomes zone of overlap. The Z to Z line get closer together because we're really pulling the sarcomere a lot shorter. So page 185 does a nice job in starting to take the sarcomere structure and add some more of the ultra anatomy. So you can see where this zone of overlap is, it's where this triad exists. If you don't know what there it is right here. The triad is 1 T-tubule and 2 terminal cisternae of the sarcoplasmic reticulum. And the terminal cisternae, basically their job is going to store calcium, because calcium is required to let actin and myosin interact with each other. You'll find out more information about that in other videos as well as lecture. So a lot of Objective 1 we just covered. The rest of Objective 1 is by an illustration on a further page, but it also answers Objective 3. So it's what is the epiperi endomysium, aponeurosis, and tendon. So if we look at page 187, you can see that the endomysium is a connective tissue structure that's around one muscle fiber. So if we look at a muscle fiber, it's got the sarcolemma around it, and then wrapped around the sarcolemma, so the sarcolemma is the plasma membrane, if you will, the muscle fiber, then we're going to have the endomysium around it. And the endomysium contains satellite cells. It's a conduit for blood vessels and nerves, or not blood vessels and nerves, but a conduit to make sure we get adequate circulation. And those myosatellite cells are really good in repair of those muscle fibers. Well, if we grab a bunch of these muscle fibers, this structure right here is called a fascicle. You saw this term in nervous system. And around this fascicle, you're going to see the perimysium. So that's going to have conduit for, blood vessels, things along that line. If you take all the fascicles and look at the whole muscle, it's surrounded by the epimysium. What's cool is that the endo, peri, and epimysium consists a lot of collagen fibers. So what we know is that this is a very strong connection. Also look how many collagen fibers are going to be within around every muscle fiber, which is surrounded by collagen of the perimycelium, which is surrounded by collagen of the epimycelium. For lack of a better term, all these collagen fibers are braided together. And if you look over here where the femur is, the muscle fibers pretty much stop here, and then the rest is all braided collagen from the endoperi and epimycelium. So that's where we get tendons from. A tendon, this is going to be a bundle of connective tissue, where in aponeurosis, this is going to be a flat sheet. So look at the dome of your head. That's where you get the Galilea aponeurosis, where your frontalis muscle attaches to. So we got objective 1-3 done. Objective, well, obviously study it. Objective 2 says, can you tell the difference between smooth skeletal and cardiac muscle? And if you go back to your lab manual, these are pages in the version that I have. You might have a different version. Sometimes you have older, sometimes you have newer. So it's within a couple pages of that page number. So can you tell the difference between them? Well, here's how you tell. So this picture right here, this is skeletal muscle. How can you tell? Striated is the first giveaway. The fibers are straight. And notice that there's a crap ton, lots of nuclei. The more nuclei you have, the more proteins that can be made. The more proteins that can be made, the faster muscle contraction. And if, I don't know, a bear is chasing you, you want lots of strong muscle contraction very quickly. Over here is cardiac muscle. When we look at cardiac, the giveaway, first off, is striated. Okay, you can't argue that one. It's pretty striated. But you'll also see branching fibers and then the intercalated discs. And as we learned in 205, when we did Unit 4 with tissues, the intercalated discs are gap junctions and desmosomes that chemically, electrically, and physically connect all cardiac cells together, which what happens to one cardiac cell would automatically means it happens to the rest. And when we get to cardiovascular, we'll actually define that out a little better. This last one down here, this is smooth muscle. And you'll see very clearly it's not striated. You'll see it's spindle shape. So you can kind of see a drawing here where, that's not very good. So this is kind of fat and it tapers at the ends. Notice this is one nucleus, and so is cardiac. Cardiac is also one nucleus. But non-striated pretty much gives it away. I forgot to mention on the previous slide, but when we look at the striations, that goes back to the sarcomere. Because remember we had thick and thin? Well, when we look at the Z line, that's going to be dark. But then you just have actin. And then you have a zone of overlap. And then you have the H zone. And then you have the M line. So notice we have these alternating light and dark bands, and that's where the striation comes from. So in other words, make sure you can identify, because you will have a question or two regarding identification, the difference between skeletal, cardiac, and smooth muscle. A reminder, as you very well, hopefully crossing fingers, you knew that you had 22 muscles of trunk and arms coming up for this lab. You will have muscles of head and neck on the next quiz, but by far the bulk will be trunk and arm. So as you were told last lab, start learning these. I really hope that some of you have already started learning these 22 muscles, how to spell them, A's, O's, and I's. If you already took the Lab 3 quiz, then you kind of know what to expect. The last one, the last objective we're going to look at today says, hey, jump to exercise 14. So if you jump to exercise 14 in your lab manual, that'll be the experiment today, looking at contribution of ATP, different ions, and looking at glycerinated muscle. So page 232 and 233, it's going to be activity 1 that this lab was supposed to happen. Unfortunately, we can't send you muscle at home to play with. I don't know why, but it has to stay really, really cold, and it doesn't do that when we ship it very well. So let me explain the experiment and then talk about what you guys are going to be doing. Basically, if you're going to read through activity 1, this gives you an idea what's going to happen. Basically, I would have given you a chunk of muscle, and we would have done an experiment with it. So the next few slides that I will show you has pictures of that, but while we're still looking at the lab manual, I want you to read all of this, okay? This is going to be questions that will be asked on your lab quiz next week. So make sure you take time to read through it and understand the experiment. Over here, this tells you how to determine percent contraction. You're going to need to know that to do your data sheet today. And your data sheet actually looks very similar to this table down here. So if we were doing this experiment, you would have done this as a group, and this is what you would have seen. This right here is a chunk of psoas rabbit muscle. It's white. We'll learn about red versus white muscle a little bit later on. There is a difference between fast and slow fibers. So you can see these are white fibers, so put that in the back of your mind for lecture. And basically what would have done is we would have given you 2 teasing needles, which basically are 2 glass, very, very fine tip probes, and you would have brought those together and pulled this muscle apart to get like individual fibers if possible. Now you can't see individual fibrils, but as small as possible. So you would have teased those apart. This is probably the longest part of the lab that you guys missed, is not, trying to dissect this apart. So then you would have put it on, as you can read through the experiment, you would have 3 microscope slides. One would be ATP, one would be salts, and then the other would be both. And you put 3 as small of fibers as you could get on each one of those slides, and then measured the length. So you can see this slide was made up, and I was trying to go quickly because we're getting kicked out of the lab, let's be honest. So this fiber actually was here, so it was even. So I would have measured this to be, 32 millimeters long. Okay, so that would have been fiber 1, and then I would measure this one, and then I would measure the length of that one. So that would have given me my starting or initial length. Okay, so this, you can't really see, but this says both, and then I would have done it for the ATP slide and the salt slide. So here you can see I've got my 3 slides. You really can't see the muscle fibers on it because they are so white, but the slides kind of fade when I put it in the black lab table. But over here you can see I've got 3 different solutions: salts, ATP, and both. And so I would have put the salt solution, I would have put a drop on there, ATP on the drop, both on there. Now I would have done it separate to allow for someone to measure. So you would have got 3 initial length. Then you would have added solution, waited like a minute, 30 seconds. Once you get to 30 seconds, it doesn't matter. And then you would have remeasured the fibers to see if they contracted or not. Okay, so somebody would have, taken that slide and remeasured the fibers again. All of which you would have put on a data sheet. So let's walk through the data sheet. Currently, the finished data sheet for this lab is not quite done yet, but I can still walk you through what you will need to do. So this first part is all based on, do you know your sarcomere anatomy? Then find your equation to calculate percent contraction. Then what will happen is this data right here will be given to you, okay? So data to be provided. So your data sheet will probably look like this when you print it out. Then there will be data posted on Blackboard. You need to fill out your table with the data. Then you need to do all the calculations. Okay, so let me, I got a little happy erasing here, or coloring here. Basically, the data you would be given are these two rows for these three columns. Same thing here. So what you need to do then is to determine the percent contraction and the average. Percent contraction, average. Percent contraction, average. So that's what you will be spending some time doing. Then, after you're done with that, make sure, to make this bigger, I didn't copy the blank space at the bottom, but there are questions down here that you need to answer based on your calculated data, okay? There are videos online, or videos in the Lab 4 folder that actually show this experiment. So this is kind of cool. There's two of them as of this moment. Go in and watch them. They're only a couple minutes long, but you actually get to see muscle contraction occur. So once you have this completed, that will be the experiment part of your lab. There are plenty of other things to do. For example, you've got 22 new muscles to learn. So when you are done with that, make sure you get your data sheet submitted on time. Please make sure you've taken quiz 3 already. And as always, if you have a question, check your syllabus, check the emails, because we do communicate a lot of information out. And if all else fails, email your lab instructor. Good luck with lab this week. Make sure that you continue to be safe and take care.